Welcome to the fascinating journey of scientific exploration, where curiosity leads to discovery and questions pave the way for innovation. This guide is your road map through the process of conducting a scientific investigation, designed to demystify the steps and terminology involved, making the world of scientific research accessible and engaging. Whether your interest lies in the biological, chemical, physical, or environmental sciences, the principles of inquiry remain the same. Let's embark on this educational adventure together. Choosing a research question. The first step in any scientific inquiry is identifying a topic that piques your interest. It's essential to narrow down this interest to a specific, testable question. This question forms the foundation of your investigation and guides every subsequent step. For example, if you're intrigued by the natural world, you might wonder how environmental factors affect plant growth. A testable question could be, how does soil pH influence the growth rate of tomato plants? In scientific terms, a testable question is one that can be answered through direct observation or experimentation. It should be specific enough that you can measure or observe changes based on different conditions or treatments you apply. Formulating a hypothesis. After choosing your question, the next step is to predict the answer. This educated guess, known as a hypothesis, is based on your current knowledge, previous research, or observations. A hypothesis might state, if soil pH levels are increased, then the growth rate of tomato plants will decrease. This prediction should be specific and falsifiable, meaning it can be proven true or false through your experiment. A hypothesis serves as a proposed explanation for an observation or phenomenon. It's a starting point for further investigation, guiding the design of your experiment by specifying what you expect to happen and why. Conducting a literature review. Before you begin experimenting, it's crucial to delve into existing research related to your question. This step, called a literature review, involves seeking out scientific articles, textbooks, and reputable online resources to understand the current knowledge about your topic. This background research helps you grasp the concepts and variables involved in your experiment and may refine your hypothesis based on what has already been discovered. The literature review compiles and synthesizes previous research on a topic, providing a comprehensive overview of what is already known and identifying gaps your experiment might fill. It ensures your investigation is grounded in scientific context, enhancing the relevance and contribution of your work. Identifying variables. Every experiment involves different types of variables. The independent variable, which you change or manipulate. The dependent variable, which you measure or observe. And controlled variables, the conditions you keep constant to ensure a t-test. For example, in an experiment to test how soil pH affects plant growth, the independent variable is the soil pH levels you adjust. The dependent variable is the growth rate of the plants, and controlled variables might include the type of plant, amount of sunlight, water, and so on. Understanding these variables is crucial for designing a controlled experiment. It allows you to isolate the effects of the independent variable on the dependent variable, ensuring that the changes you observe are due to your manipulation and not other factors. Conducting a risk assessment. Safety is a cornerstone of any scientific investigation, ensuring not only the protection of the investigator, but also the environment. A comprehensive risk assessment involves 3 critical steps: identifying potential hazards, assessing the likelihood and severity of these risks, and implementing controls to mitigate them. Here, we illustrate this process using a specific example related to the use of fertilizers in experiments. Identify. A notable risk in experiments involving fertilizers is the presence of heavy metals, which can pose significant health risks. These substances may cause reproductive problems, birth defects, and cancer if inhaled or ingested. Identifying this risk is the first step in safeguarding against potential harm. Assess. Upon identifying a potential hazard, the next step is to evaluate the risk it poses. In the case of heavy metals in fertilizers, we judge this as a medium risk. However, most risks will likely be rated as low. This assessment considers both the likelihood of exposure and the impact such exposure would have on health. It's a critical step in understanding the scope of precautionary measures needed. Control. The final step focuses on implementing strategies to minimize the identified risk. When handling fertilizers known to contain heavy metals, wearing protective gear such as face masks, gloves, and goggles is imperative. These controls are effective in reducing the risk of inhalation or ingestion of harmful substances. Additionally, washing hands thoroughly after use is a simple, yet crucial practice to further mitigate risk. This structured approach to risk assessment ensures that all potential hazards are systematically identified, evaluated, and controlled. By applying these principles to your own experiments, you can maintain a safe and productive research environment, enabling you to focus on the scientific inquiry at hand. Collecting and analyzing data. With your experiment underway, it's time to collect data. This involves making careful observations and measurements related to your dependent variable. Systematic data collection is critical for analyzing your results. Once collected, your data can be organized into tables or graphs, making it easier to identify trends, patterns, or differences between your experimental groups. Analyzing this data often involves statistical methods to determine whether the changes observed are significant or could have occurred by chance. This analysis provides evidence to support or refute your hypothesis, contributing to the body of knowledge on your topic. Discussing your experiment. After analyzing your data, reflect on the experiment's process and outcomes. Evaluate the reliability, validity, and any errors or limitations of your study. Discussing these aspects critically examines the strength of your scientific inquiry, addressing questions such as how consistently your experiment can be replicated and whether it accurately tested what you intended. This discussion should also propose future research directions based on your findings. It's an opportunity to suggest how your investigation could be extended or improved, sparking further inquiry and exploration. Concluding your findings. The final step is to draw conclusions from your experiment. Summarize the outcomes, stating whether your data supported your hypothesis. Reflect on what you've learned and how it contributes to understanding the problem you investigated. This conclusion ties your investigation back to the original question, highlighting the significance of your findings within the broader scientific context. By conducting your own scientific investigation, you not only contribute to our understanding of the world, but also develop critical thinking, problem-solving, and analytical skills. This process, from posing a question to drawing conclusions, embodies the essence of scientific inquiry, a cycle of curiosity, exploration, and discovery that fuels the advancement of knowledge.